I'm Melissa Parker. I work at Insight, and I'm here to present ruxolitinib cream ameliorates preclinical model of skin dermatitis. Closer. Is that close enough? Can you hear me? Sort of. Yeah. Okay. So we're trying to look at contact dermatitis and the treatment of ruxolitinib. And I just wanted to highlight, there's a point here. So we're trying to look at a Th2 model-driven contact dermatitis. We use the FITC Th2 haptogen in a preclinical mouse model. And we're evaluating ruxolitinib, it's a JAK1, JAK2 inhibitor that we've seen efficacy in previous clinical trials in atopic dermatitis. So you can see here, which the Th2 response involves itching, cytokine release, which results more scratching, barrier dysfunction, and back to Th2 skewing. So it's a cycle that, with the itch-scratch cycle, that resolves in more Th2 and cytokine response. And for the JAK1, JAK2 relationship, the Th2 cells secrete IL-4, bind to IL-4 receptors, which signal through JAK1, JAK2, and, sort of Th2 differentiation. Oops, sorry, my bad. Yeah, there we go. Alright, so for our method, we used a chronic FITC skin dermatitis model. So we did an initial sensitization with the FITC on the abdomen, and then we did every 7 days elicitation ear challenge. We monitored the weights bi-weekly. We did ear swelling bi-weekly, and at the end we collected auricular lymph nodes, draining lymph nodes, and the ear for RNA analysis. Okay, so you can see that the model was well tolerated by the mice. The vehicle didn't show any kind of severe drop in body weights, and you can see that our treatment with BID and QD had very stable body weights, and with the steroid cream we had significant weight loss, which you would expect. And as far as the actual driven disease, we can see in the vehicle group the ear swelling and the contact dermatitis did push the disease, and it was controlled nicely by ruxolitinib cream as well as the steroid. Okay. So then we did some immunophenotyping in the draining lymph nodes to see what kind of a cell population is there. And so we did a total cell count, and as you would expect with the ear swelling, we saw a much higher total cell count in the vehicle group and a much lower cell count in the treatment, the BID treatment and the betamethasone, but you do see a dose dependent in the once daily Rux treatment. When we broke the T-cell down for immunophenotyping, we see Th1 cells, you don't see any difference in the once daily but twice daily. We see, but it is statistically different from the vehicle group. And then we see Th2 have a nice statistical difference with the twice daily, but not with the once daily. I can barely hear myself. And as expected, with the Th17, we're not seeing any differences, which we would hope not with the JAK1, JAK2.
I didn't hear myself. It's loud.
Okay.
You have not analyzed the steroid here.
Oh, yes. So we did not, because the steroid works through a completely different mechanism, we didn't actually bother to, have that here because the mechanism is so different. It's not through a Th1, Th2. So it's just used as control. Okay. So then we did RNA analysis via NanoString. I did 770 genes for the treated ear, and you can see how they group very nicely with the vehicle, once daily, twice daily, and the steroid cream. You can see a very striking difference between the vehicle and what we got treated, and also you can see a mechanistic difference between our JAK1, JAK2 inhibitors and the steroid cream. So it's good with good inter-animal consistency and a nice difference in the pathway profiling. God. Okay. So we just wanted to highlight, this is the BID versus vehicle, and so there was about 60 genes that were over log 2-fold with a statistical significance via this analysis. And so you see a very large pathway difference you can highlight. And so we just wanted to highlight some of them where the immune activation, IL-4 that you'd expect, some T-cell activation, Th2 relevant, chemokine signaling, and then also since we did a JAK inhibitor, we also can see we saw some differences in the JAK pathway. SAT1 interferons, and then JAK3 STAT1.
Can you say a bit about CCL8, because that's by far the strongest change that I can see?
Right. So we actually haven't had a chance to really get into that too much in CCL8, but our thoughts are that with just a chemokine signal, that it's just driving either the inflammatory cells in and out of the areas. Those down signals, so...
Yeah, because it's more than 8-fold change. That's quite a lot. It's highly significant.
Yeah.
Quite interesting.
Yeah, we're still working on trying to do a little more breakdown of this pathway analysis and determine exactly what we find and how much of a signature we have in there. So anyway, in conclusion, I just wanted to, in conclusion, we see a mouse model of skin dermatitis with Th2 and a good control with our twice daily Rux cream. And yeah, thank you very much.
Thank you very much. Are there questions from the audience? Do you have any plans to do a comparable study in humans? Because you would be able probably to do the same thing.
Well, so right now we're looking at the Rux cream in atopic dermatitis, and then we're trying to see how that goes, and then potentially
Including mRNA profiling probably, right?
Correct. And I do have another poster in there in the exhibit hall regarding Rux cream use on atopic dermatitis.
Did you also screen the mice for any safety things, safety signals associated with the application of the cream?
So we didn't do that in this study. We've used this Rux cream in a lot of the mouse models, and we haven't seen any issues, but this has been used in a lot of preclinical models without issue.